I hope my slide is visible and my voice is audible. Is it fine?
Yes, sir.
Okay, then we will move on to today's session. Once again, welcome you to all the course Environmental Biotechnology for the course code of NOC 25BT83. So this is going to be the week 10 live interaction session on the course Environmental Biotechnology. And myself, Ajit Kumar, a PhD research scholar from the Department of Bioscience and Bioengineering from IIT Guwahati. So this week we will see first the week 10 recap of the lectures and their important concepts, followed by the sample MCQs. So this is going to be the today's session overview. So without any delay, we can move on to the course. Before that, I will personally introduce about myself. Myself, Ajit Kumar, a joint degree research scholar from IIT Guwahati and IIT BHU Varanasi, and I come from the laboratory of biochemical and environmental engineering, where my lab works on wastewater treatment in different industries, and my research inclines to the application of enzyme in wastewater treatment, especially the textile wastewater treatment. And moving on to my the qualification, I have done BTech Biotechnology from Shastra University and MTech Industrial Biotechnology from NITK Surathkal, Karnataka. Moving on to my work experience, I worked in many pharma companies like Biocon and Enzene Bioscience. So this is all about my personal introduction. So without any delay, we will move on to the week 10 in live interaction session. And now we will see the recap of the week 10 lectures. So before that, we will just recap what are all the things that we have seen in the week 9. In the week 9, we have started with the application point of view of the environmental biotechnology course, where we have seen the first application with this bioremediation. And we have seen what is bioremediation, and we have seen the evolution of bioremediation from 1900 to present. Then we slowly move on to the need of bioremediation in microbial ecology. Then we have seen the classes of pollutant, what are all the different classes that are prevalent in the environment. Among that, we have seen the most important concept of emerging contaminants and why it is called an emerging contaminants and the examples and day-to-day examples in different industries we have seen. And we have also seen the importance of microbes, that is using the microbes in the bioremediation. Why microbes especially? Then we have seen the advantages and disadvantages of bioremediation, followed by different types of bioremediation, which is in situ, intrinsic bioremediation, engineering bioremediation, and ex situ bioremediation. So these are all the types of bioremediation that we have seen in the last class, along with the examples. Then moving on to the approaches of bioremediation, we have seen some important concepts like biostimulation and why it is important and what are all the examples we have seen. And we have seen the bioargumentation along with the one day-to-day example. Then we have also seen phytoremediation, and we come to know that not only microorganism, also plants also helps in remediating the environment, and that's why we call it as phytoremediation. Then at last, we have seen the challenges of bioremediation in microbial ecology. So these are all the topics that have been covered in the last session. And if you have missed out, you can revisit those videos and get uh, more info on that. So in this bioremediation, we will continue in this session, we will continue on the topic bioremediation and we will see some more important concepts regarding this. So at last class, we have seen bioremediation concept where what is bioremediation? It is nothing but it is a managed or spontaneous process where the term itself denotes we are going to use the biological catalyst. Biological catalysis means it can be microbes. Microbes can be prokaryotic or eukaryotic. It can be fungi, yeast, protozoa, etc. And not only microbes, it can also extend to plants and insects too. So by using the biological source or their enzymes to act on the pollutant, and we have also seen several classification of pollutant. So these biological catalysis acts on the pollutant to remediate or to eliminate these contaminants that are present in what environment. It can be water, it can be wastewater, it can be sludge, soil, aquifer material, or gas stream. So, in short, the bioremediation refers to cleaning up the environment using biological source. We call this term as bioremediation. And how does it helps? It helps to reduce, eliminate, or contain these hazardous contaminants by not entering into the environment. And what are all the key players that involve in this remediation? As we have already seen, especially the microorganism, it can be bacteria, it can be fungi, it can be yeast, it can be protozoa, anything. And it also extends to plants and insects to transform and degrade these type of pollutant. And what are all the important factors that determines the progress of the bioremediation? The main important factor is that physical and chemical environment, which includes it can be temperature, pH or oxygen level. And also it depends upon the microbial community that are present in the particular environment. And third important factor is the nature of contaminant, which denotes the pollutant nature. It can be antibiotic in case of pharmaceutical pollutant and dyes in case of textile pollutant and pesticides in case of agricultural sector. So depending upon the nature of the contaminant, depending upon the microbial community and also the physical and chemical environment, the transformation and degradation of the bioremediation changes from site to site. So this is the broad recap of the bioremediation. Now we will move slowly to the concepts of point source and non-point source. The term, the term itself tells us that the point source, that is there, these contaminants are easier to find the origin of pollution. You can directly pinpoint where the pollutant is coming from. For example, if you see in the right hand side of the diagram, the factory draining pipe pouring the waste into the nearby water bodies, that is nearby river. So as we understand that the wastewater is directly coming from the source of place, which is the factory. That's why we call it as point source. That is, we know the origin of pollutant. That is here in this case, it is factory. Similarly, if the pollutant or the wastewater releasing from the wastewater treatment plant, this is also comes under the point solution. That is, these are all single point of contaminants. These are all single point of contaminant. That is, their source of pollutant is well known. In this case, it is factory, and in this case, it is wastewater treatment plant. So, what happens? The point source is nothing but the source of contaminant can be easily find out. So you can directly point where the pollutant is coming from. Whereas in non-point source, it is like an hidden pollutant. What do you mean hidden pollutant? For example, During rainwater, the washing of dirt from the roads into river, so or from the crop yield or from the city streets, the wastewater is accumulating and finally it is entering into the river. So in this case, we can't pinpoint the pollutant in one place. So it comes from different direction. So the source of pollutant cannot be determined as easy in case of point source. So that is why it is called as non-point source. For example, if you see in the pesticide, we spray pesticides on the crops and fertilizers from the farm. So what happens when the rainfall comes? When rainfall comes, it collects all the pollutant and carries them into the nearby river or lake. So here we cannot pinpoint the source of pollutant where its origin is. So in short, to summarize, the point source is nothing but it is easier to identify the origin of pollutant. Whereas in non-point source, the origin of pollutant site cannot be determined as it is. So it needs much effort to determine the origin. So why we need to understand this point source and non-point source? Because this determines the what strategy can we infer to a particular pollutant. So this plays an important concept knowing the point and non-point sources. So if you see the nature of the pollutant, so it depending on their behavior of the pollutant, the organic compound, that is organic compound is nothing but it is mainly composed of carbon compounds. So these are classified as first is biodegradable, second is persistent, and third is recalcitrant. So what do you mean by first term, the biodegradable? What happens here is first, in biodegradable, the microorganism consumes this organic compound and transform them into less toxic, which is CO2 and H2O. So normally what happens during biodegradation, they do not cause any trouble. So they easily undergo the biological transformation. That is, the microorganism will be consuming this organic pollutant and they transform into less toxic or non-toxic form. Whereas in the case of persistent compound, we call this as stubborn carbon. That is, they don't leave easily. They stick to the environment for long period of time. For example, we can say the pesticides and pharma contaminants. They don't degrade easily. And these organic compounds, they do not undergo the bioremediation process in certain environment. That is why they persistent. They are persistent, they are stubborn. Whereas recalcitrant, it is like unwanted guest. That is, they always resist the degradation in almost all the environment. So this is very hard to degrade. Otherwise, it is very resistant to the biodegradation. For example, you can take the plastics or polychlorinated biphenyls. So these are complex and recalcitrant in nature. They are complex and recalcitrant in nature. That is, they often resist these biodegradation. So these are all the three important terms that you need to be remember while understanding the nature of the pollutant. And among this, if you see the biodegradable is easier to degrade, whereas the persistent and recalcitrant, depending upon the environment, they are difficult to degrade and they often resist the bioremediation. So understanding the organic compound and keeping in the particular category helps us to strategize the approach of bioremediation in the wastewater. So first goal is to you have to identify the organic compound. The first aim is to identify the organic compound. whether it is biodegradable, whether it is a persistent or recalcitrant. After analyzing the organic compound, you can next step is to determine what strategy can we employ in order to degrade the particular compound. So if it isn't biodegradable, you can straight away go to the microbial based degradation. Whereas if it isn't recalcitrant or persistent, you have to adapt a strategy that a combined approach of physical, chemical, and biological way of treatment in order to completely mineralize them. So it is very important to understand the nature of pollutants that are present in the environment. So moving on to the important definitions that we come across normally in bioremediation is that the first is biotransformation. As the term itself denotes that the microorganisms are used to transform the complex organic compound into less toxic or simple compounds. That is, the microorganism first will consume this organic compound and they oxidize into CO2 and H2O. This we call it as biotransformation. That is, they try to reduce the complexity of the organic compound so that the chemical structure has been completely changed or reduced to non-toxic form. So what happens? The environmental engineers and scientists, they often use this term to signify the alteration of the molecular structure. For example, the chlorinated biphenyls are very complex and they contain multi-benzene rings. So the biotransformation indicates that there will be alteration in the molecular structure of the compound or there is a change in the oxidation state by using the microorganism or their derived enzymes. So if you are able to alter the molecular structure, we call this term as biotransformation. So such alteration or such changes in the molecular structure, sometimes it helps in decreasing the complexity of the intramolecular bonds, but sometimes it can also increase the complexity of the molecule. So depending upon the process and the microorganism metabolic pathway function, it can decrease the complexity or it can sometimes increase the complexity. For example, if you see the benzene compound, sometimes it tends to increase the complexity by binding all the benzene to form multi-benzene compounds, which is toxic in some cases. So, depending upon the microorganism and their metabolic pathway, this transformation occurs. So, the next important term is that biodegradation. As the term itself suggests that degradation of organic compound occurs using the biological source. Here what happens? Here there will be reduction in the complexity of the molecular structure of organic compound by breaking their molecular bond. It can be hydrogen bonding, van der Waals force, or covalent bonding, etc., which ultimately results in simpler structure. So generally, the microorganism will consume these organic compound and they oxidize into simple and non-toxic form by breaking their important bonds. So here what happens? There is a substitution functional group occurs or they try to break their functional group, which results in complete breakdown of this organic compound. So this is the important concept or definition that you have to remember. First is biotransformation, where using the microorganism, we tend to transform the chemical structure of one organic compound to the other organic compound, mostly simple structures from the complicated structure. Whereas in biodegradation, we tend to degrade these compounds to the simple and non-toxic form. So these are all the two important factors, and you need to understand the differentiate between these two terms. Then these biodegradation of most priority pollutant, which is emerging pollutant, they often results in lesser toxicity. That is, through biodegradation, what we get lesser non-toxic products, and we call this process also called as detoxification. That is, removing the toxic behavior of the organic compound using the microorganism. But sometimes what happens is the pollutant may be transformed into an another product that might be have a toxicity behavior. So this transformation, which helps or which increases the toxic nature of the compound, we call this as activation. So it activates the compound to greater toxicity than their parent compound. So these are all some of the important consideration you have to keep in mind while assessing the risk that are associated with the bioremediation process. So you can biotransform it or you can biodegrade it or sometimes you can biomineralize it completely. So this we will be seeing in the upcoming slides. Before that we will see one example of activation where it includes by reductive dechlorination, that is removing the chlorine compounds of the solvent trichloroethylene to vinyl chloride under anaerobic condition. So here what we are trying to achieve, we are trying to convert an complex molecule which is trichloroethylene to simpler molecule like vinyl chloride under the anaerobic condition. So this is one of the example for activation where complex substance is converted into simpler compound. And the other example is epooxidation, that is oxidizing the pesticide. For example, one of the pesticide is aldrin to dialdrin under aerobic condition. In this case, what happens? The compound is transformed into another compound by merging two aldrin group becoming a dialdrin, which increases the toxic behavior of the particular compound. So these are all the some of the examples for biodegradation. Sometimes it can help in degrading the complex, but sometimes it can also increase the toxicity. So that is why you have to, before selecting any bioremediation technique, you have to see their risk assessment also. So, as I said, we have seen the biodegradation, biotransformation, and the most important concept is next is the biomineralization. The term mineralization itself denotes that the organic compound is completely mineralized. To what? Into it can be CO2, CH4, H2O. So these are all the simple and non-toxic in nature. So what happens here? Here also biologically catalyzed. It can be a microorganism or it can be an enzyme which helps in degrading this organic compound and helps in converting into mineralized compounds like CO2, CH4 or H2O. So in general what happens? In general, the mineralization inside the microbial cell, they always undergo a special catabolite pathway which process these organic compounds and try to break down and get energy from those compounds. That is, they can sometimes they can use these compounds as a carbon source to grow the cells and for microbial activity. And why it is very important to learn this biomineralization concept, because it helps in achieving the final cleanup stage. That is, here what happens, the complete mineralization of compounds is occurring into CO2, CH4 and H2. So as compared to the biodegradation and biotransformation, the biomineralization is a powerful strategy which helps in completely wiping out the pollutant into safer and natural form. So this is why biomedialization is always the ultimate goal for the bioremediation of environmental pollutant. So till this, we have seen the important terminologies and what is bioremediation, and we have seen terminologies like biodegradation, transformation, mineralization. So these are all the techniques that helps to achieve the environment against the pollutant. And next concept is factor affecting the bioremediation, where the first important concept is concentration of the pollutant, or we call it as an contaminant. Why concentration is more important in determining the bioremediation? So let's see one example. So we will take one day-to-day example. So you can think of the sugar that we put in the tea. So what happens if the sugar is too little? We hardly taste it, right? We cannot feel the taste of tea. In other another case, if the sugar is too much, what happens? The tea became unpleasant. Similarly, in microorganism also, if the concentration of the pollutant, if it is present at very low concentration, the microorganism don't bother to produce appropriate degrading enzymes. But what happens if the concentration is very high? If the concentration of the pollutant is very high, it acts as a poison to the microbes and finally it inhibits the microbial growth. So this tells us that the concentration is directly affects the microbial activity. So if it is lowest, it doesn't trigger the degrading enzymes. Whereas if it is very high, it might feel too toxic to the microorganism and finally inhibits the microbial growth. So this is why an optimum concentration need to be found out to determine the progress of bioremediation. And if you see the decomposition rate of this catabolic enzymes, it can also be increased by the synergistic interaction between various components of the contaminants. So this is why the concentration of the contaminant is very important while studying the bioremediation technique. Since it is directly affecting the microbial activity, we have to optimize this factor to which level the particular microorganism can handle the contaminant. Because in case of dyes, the microorganism can deal with some 100, 200, even 500 ppm of dyes. But the same microorganism cannot handle the heavy metal presence, even in the millimolar level. So depending upon the pollutant and their concentration, it affects the bioremediation approach. And the second important factor that affects the bioremediation is nutrient availability. And why nutrient availability is so much important? Because every microorganism needs food, and the food can be carbon source, nitrogen source, phosphorus, potassium, or calcium. Why it is very important? Because they are important for their growth of microorganism, and second importantly, their concentration of the nutrient is also determines the degradation efficiency. So in case of nutrient availability, if they get the right balance of nutrients, their growth can be effectively increased and it can effectively in turn degrade the bioremediation of pollutant. What happens if the amount is too much? For example, if there is an too much level of nitrogen and phosphorus, it is going to disturb the whole metabolism. It is like overeating the junk food. What happens if we overeat the junk food? It leads to obesity, cholesterol, blood pressure, etc. Right? So, for microbes also, we have to find the balanced nutrient in order to improve their activity, microbial activity, which in turn helps in degrading the pollutant. And third important point is that the rate of bioremediation is also determined by the accessibility of the particular pollutant towards the microorganism. So what happens? You have to make sure that the pollutant is rightly available for the microbes. So the we call this term as bioavailability. So how much pollutant is available to the particular microorganism. We call this at bioavailability. More the bioavailability, more pollutant will be up. Reaching the microorganism is very high, so it increases the degradation efficiency. So this is why the nutrient availability also determines the biodegradation of the pollutant. And third is surfactant. So what happens? The main role of the surfactant is to increase this bioavailability. As we seen in the previous slide, the importance of bioavailability. That is how much pollutant is available to the microorganism. So in order to increase their bioavailability, surfactant play an important role in enhancing the bioavailability of the particular pollutant to the microorganism. So mostly what kind of bio surfactants are used to increase the bioavailability? One is chemical based surfactant and second is food grade surfactant. So these are all types of surfactant that are used to increase the hydrophobic organic contaminant. And if you see the chemical based surfactant, we have examples like Triton X100, Twin 80, and SDS. So these are all some of the chemical based surfactant, and these are all petroleum derived chemical surfactant, which enhances the bioavailability of the pollutant. Whereas in case of food-based, food-graded surfactant, we have TMAS 28, TMAS 10, and TMAS 60. So these are all the some of the examples of surfactant. And moreover, beside chemical and food-grade surfactant, now the surfactant is also produced by the microbes. So what happens? It helps in mixing the pollutant with the water so that microorganism can be attached to it and degrade efficiently. So surfactant play an important role in determining the bioremediation, and moreover we have three types of surfactant. One is chemical based surfactant, one is food grade based surfactant, and third is biosurfactant. So these are all the surfactant and the importance of surfactant in the bioremediation. And fourth important factor is that the characteristics of the contaminated soil. So the bioremediation process is significantly affected by different parameters such as pH, texture, permeability, water holding capacity, temperature, and oxygen availability. Among this, we will see some factors here. The first is the pH. So, what happens? Mostly, microorganisms prefer the optimum pH. What do you mean by optimum pH? Optimum pH is the pH where the microorganism growth is higher. So, optimum pH is often preferred for the bioremediation process, and it prefers neutral pH, that is 6 to 8, especially for degrading the petroleum hydrocarbons. Whereas if you see for some fungi or acidophilic microbes, they can degrade the pollutant effectively at the acidic range of environment. So depending upon the organism, different pH is required in order to increase the rate of degradation. And the second most important environmental factor is that the temperature. The degradation of the contaminant is also affected by the temperature, especially in case of hydrocarbons, where we carry out in situ and ex situ conditions. And it has been found that an optimum temperature of 30 to 40 degrees Celsius increases the bioremediation in the soil as well as the marine environment. What happens if the temperature is very low? If the temperature is very low, it slows down the microbial growth. Whereas if it is too high, it kills the microorganism, which is going to drastically affect the rate of biodegradation. So here also, optimum temperature needs to be found out for the particular microorganism in order to carry out the bioremediation process. Whereas the third important factor, which is the oxygen availability. What happens? Oxygen is the very important factor to determine the rate of biodegradation of the contaminants. And especially, there are two types of oxygen availability. One is aerobic mode of degradation and the anaerobic mode of degradation. And as we already know, the aerobic mode of degradation is nothing but the process that carry out in the presence of oxygen. And anaerobic processes, the biodegradation happens in the anaerobic condition, that is in the absence of oxygen. And from the earlier lectures, we understand that aerobic biodegradation is much faster than the anaerobic biodegradation. Why it is so? Because here oxygen is used as an electron acceptor, and this is going to yield an higher energy for the compound in oxidizing the organic compound. And if you see for the aerobic respiratory breakdown of organic contaminant, oxygen is going to be the key factor in determining the rate of extent of biodegradation. Whereas in case of anaerobic mode of degradation, we have other options like nitrates and sulfates, which play an important role and acts as an electron acceptor to extend the rate of degradation. And in majority of the cases, they are adding the hydrogen peroxide to introduce the oxygen externally. And we know that oxygen peroxide is 7 times more soluble than the oxygen, especially in the water. So, oxygen availability is one of the key factor and they are the biggest driver for the bioremediation. And this can act in the presence or absence of oxygen. So, these are all the three important environmental factors, which is pH. We always tend to have the optimum pH, temperature. We always tend to have the optimal temperature. And third point is oxygen availability, depending upon the mode of respiration, that is aerobic mode or anaerobic mode, we tend to provide the oxygen separately. So these are all the environmental factor and these are all the uh, chemical factors. And first is concentration of the contaminant. Next is the nutrient availability. Third is the presence of surfactant. So these are all the most important factors that governs the rate of the bioremediation. Then we will move on to how these microbes interact with the organic compound. In the earlier lectures, we have discussed a lot of strategies. One is biodegradation, bioaccumulation, bioabsorption, bioprecipitation. So in this session, we will see more onto it with examples. The first is biodegradation, which is nothing but which is an biologically catalyzed substance which helps in reducing the complexity of the organic compound. And why it is so important? Because this is one of the most favored and sustainable means of removing the contaminants from the pollutant site. And the indigenous microorganism often exploits the biodegradation of many different types of organic compound as a metabolic niche to obtain the energy from the compound by degrading the organic compound, that is oxidizing their organic compound and building the synthesis of new cellular material, that is the microorganism first they will consume these organic compound and they oxidizes these organic compound and they get energy out of it. Along with that, they also tend to synthesize new cellular material, that is they also tend to produce biomass too. So that is why biodegradation is a more favored option for choosing the environmental pollutant. So here we will see the example. So what happens? The microorganism will try to oxidize the organic pollutant. They try to oxidize the organic pollutant to CO2 or H2O, and we call this process as biomineralization. Simultaneously, what happens here is the electron transports acceptors like oxygen in the case of aerobic, whereas in case of anaerobic, nitrates, iron 3 or sulfates will act as an electron acceptor and they try to achieve this degradation. So these are all the some of the oxidizable pollutant. And if you see the petroleum hydrocarbons, examples benzene and toluene, chlorinated hydrocarbons, vinyl chloride and dichloroethane, and a micropollutant which contains the pesticide, which is this is the structure of atrazine, and one of the pharmaceutical pollutant like chlorphenol. So these are all the emerging contaminants, and these are all the petroleum hydrocarbons. So what happens first? The microorganism will be oxidizing these oxidizable pollutant into CO2. Meanwhile, they releases the electrons out of it, which helps us to converting the oxygen into water in case of aerobic respiration. Whereas in the case of anaerobic respiration, if the nitrate containing group, nitrate containing environment, it will try to convert into nitrogen. If it is MnO2, it will try to reduce them into Mn2 plus, etc. So this is the hierarchy of decreasing redox potential, and these concepts we have already discussed elaborately in the week previous weeks. You can go through that. So what happens in general is the microorganism will oxidize this pollutant and convert into CO2, and they release the electrons, and these electrons will be captured by the electron acceptors and they convert into non-toxic form. So what happens to this pollutant? For example, we will take the chlorinated hydrocarbon, which is one of the important chlorinated hydrocarbon is trichloroethylene. That is due to the presence of three chlorine atoms. This is regarded as trichloroethane. And what happens during the biodegradation? This trichloro first it will be converting into dichloroethylene, then it will be converting into monochloroethylene, which is called as vinyl chloride. So upon degradation, it will be finally converting into ethylene. So what happens here is the more complex organic compound is finally converting into less toxic or less complex molecule like ethylene. And if you see in the case of nitroaromatics, which is TNT, trinitrotoluene, due to the presence of three nitrogen groups, nitrate, NO2, nitro groups to the toluene compound, we call it as trinitrotoluene. And upon degradation, it has been converting into triaminotoluene. So in this case, we are trying to reduce the toxicity by converting the nitro into amino group. So this is how the microorganism tend to reduce the pollutant or mineralize them completely. So the important factor in determining the rate and extent of degradation is that first the type of microbial community that are present in the environment. What kind of microorganism that are present in a particular environment determines the rate and extent of degradation. And next is the environmental condition, whether it has appropriate temperature, that is optimum temperature is there or not, or optimum pH, and whether oxygen availability is sufficient or not. So these all decides the rate and extent of biodegradation. So first is microbial community and second is the environmental condition. So these are is the broad overview mechanism of biodegradation and their important factors in determining the rate and extent of biodegradation. So so we will move on to the prerequisite for the microbial degradation. So as we have already understood the concept of biodegradation, which is nothing but it is a self purification process in a polluted medium. That is, it will try to eliminate the wastewater in the treatment plant, or it will try to remediate the soil or groundwater that are contaminated by the underground storage tank. So it is like a self purification process as we see in the aquaguards in our day-to-day life. And these requires conducive condition. That is, they need two basic criteria. You need to use the appropriate microorganism which has the tendency to produce specific degradase, and and the degradase should be functioning well in order to break down this complex compound into simpler structure. So If you have the correct growth and correct proportion of enzyme with correct environment, then the organic pollutant can be converted into simpler compound. That is, biomineralization can be achieved so easily, that is forming the carbon dioxide and water. So the most important condition is you need to have an appropriate microorganism and appropriate enzyme production with a suitable environmental condition. So this determines the level of degradation. So we will see what are all the requirements that are needed for microbial degradation. The first is the existence of the organism with required degradation potential. So what happens here? You need to find the right microbes to degrade or break down the certain pollutant. It can be any kind of pollutant. The first important criteria is to find the appropriate microbes for the particular microorganism. So, how it works? So, the microorganism degrades the pollutant only if they have enough enzyme that fit into their metabolic machinery pathway, that is their natural energy pathway. So, you need to identify the microorganism that has the capability to produce the appropriate enzymes. For example, we will compare two examples. First is if you are having a sugar, what happens? The microorganism will readily assimilate this sugar and they break down and they generate as much as energy possible. Whereas if you take the plastic or chlorinated solvent, which is often hazardous, more complex chemical compound when compared to the sugar. So what happens? Why it is a complex sugar? Since they contain unusual groups like unusual groups, so it often triggers the resistance or recalcitrant behavior to the biodegradation. So this is why you need to find the right microbe for degrading the pollutant. And if you observe the pollutant nature, if you have the greater the differences in the chemical structure between the target pollutant and the microorganism, the less likely that contaminant will be recognized as the substrate. So what does it tell us? If the chemical structure, if the chemical structure is so complex or more complex, the microorganism will always regard as a foreign and they always fail to recognize as their carbon source and they do not degrade the contaminant. That is why, as we see the example, the plastic or the chlorinated solvents. That is why it is a complex substance. As I told, it contains unusual groups like Cl, NO2, Cn, or SO3 groups. So this is not a normal part of biological system, and we call these substances xenophores since they contain unusual group or they are not common in nature. So these are all man-made pollutant or man-made organic compounds. So what happens? These xenophores, as they are man-made, they often confuses the microbes. So that what happens? It makes the pollutant recalcitrant to the microbial degradation. That is, if so what happens here is these xenophores are always recalcitrant in nature to the microbial degradation. So by this we understand that organism must exist with some enzymatic machinery to recognize and attack the pollutant. So scientists what they will do is they always try to find the right key. That is for the particular pollutant, the scientist will always try to find the particular microorganism in order to deal with. So that is you have to find the right key for the right pollutant. So you are the first and basic need is you need to find the right microbe for the right pollutant. So this is what the first principle tells us, that is the existence of organism with required degradation potential. So you need to find the right microbes for the particular pollutant to degrade or break down them. And from this, scientist has come up with an hypothesis of microbial infallibility hypothesis. And what does this hypothesis tell? For almost any organic compound that are available on the earth, there is always a microorganism that can break down if it is given a right suitable environmental condition. And also, if no such organic or organism exists, what happens? The evolution and adaptation will eventually create one within a short period of time. So, what happens for every organic compound that exists in the earth? There will be one microorganism in the earth that will help in degrading these organic compound. If there is no organism exist, what happens during the evolution through adaptation? The the microorganism will evolve in the direction so that the organic compound will be decomposed completely. So if I will give an instance. For instance, you can take oil spill. At first, it looks like an ecological disaster, and but soon what happens here is scientists tried to uh, get a specific bacteria. The bacteria is alkanovirus. So it emerges, this kind of specialized bacteria emerges and start to consume these petroleum hydrocarbons, and they start to degrade these oil spills that are present in the ocean. So this is one example where these species try to evolve during the evolution in order to degrade the recalcitrant pollutant. Therefore, the microbes are the great recycler of the earth. They may take time, but if they are given the right environmental condition, they will degrade as much as possible. So this is what the microbial infallibility hypothesis is saying, that somewhere or other some microorganism will exist, and if they are given a suitable condition, it can oxidize as any organic compound which is theoretically capable of being oxidized. So for every organic compound, there is at least one microorganism present in the environment to degrade them completely. So this is the first principle. And second principle is that the presence of specific degrader in the contaminated environment. So there should be always a specific microbial degrader in the pollution cleanup. So what happens? So here you can think of like the microorganism acts like a chef. So what happens? They cook and eat or taste their certain dishes, right? So likewise, in the polluted environment, the microorganism acts as a degrader. So they trained to evolve and evaluate the feed on certain contaminants. For example, we can take the example of hydrocarbons in natural. What are all the hydrocarbons that are present in nature? It can be oil, gasolines, or coals, and they have been around the earth for billions of years. And over these millions of years, what happens? The microorganisms are constantly exposing their presence to the hydrocarbons, and they evolve, and now they have the capability to utilize these hydrocarbons as their food and try to degrade them as much as possible. So this is one example with regard to their hydrocarbons that are present in their nature. And second example is that the BTEX, which is benzene, toluene, ethyl benzene, and xylene, where in 1908 scientists discovered an bacteria which is Bacillus exovorum, They observed that this bacillus can grow and consume the carbon source as toluene and xylene in the presence of oxygen. So what happens? They not only consume, they tend to degrade and mineralize them completely. So, so this is why you have to find the specific grader for a particular environment. If it is an hydrocarbon or oil spill, you have to use a particular environment or particular microorganism to degrade the particular pollutant. So this is the second principle, which is specific microbial degrader needs to be used for complete pollutant cleanup. And third principle is there accessibility. We call this as bioavailability of the target pollutant to the microorganism. Accessibility means the microorganism are ready to eat these pollutant, but unless these pollutants are available or dissolve in water or they diffuse to the tiny pores or release from the soil particle, they can't supply and get them right. So the bioavailability is one of the important factor for making availability of pollutant and making more contact of pollutant to the microorganism. And the target pollutant must be accessible in various aspects. And these, the rate of biodegradation is mainly controlled by the diffusion of this pollutant or desorption or dissolution rate. So what happens here also, we can say we can take the oil spill example where the presence of oil droplet in the water, where for example if some oil spill occur in ocean, oil mostly floats on the water as a big droplet and microorganism cannot directly consume these droplets. So only small amount of oil will be dissolving into water and this small amount of oil can only be degraded by the microorganism. So in this case, what you can do is you can introduce the biosurfactant. As we have seen earlier, there is chemical or food grade or biosurfactant available in order to increase the bioavailability. So this is one example of oil spill. And second example is the pollutant present in the nanopores. That is the pollutant in the rock or soil behaves and takes time to diffuse out before the microorganism can access to them. So from this we understood that the success of the bioremediation process not only depends upon selecting the right microbes at the right environment, but also depends upon the accessibility of the pollutant to the microorganism. That is why the biosurfactant play an important role to accumulate these pollutants and degrade them effectively. So accessibility is also another important factor which determines the rate of biodegradation. And fourth important principle is that the induction of appropriate degradative enzymes. So selecting the microbe is first thing, and second is you have to induce these enzymes in order to enhance the degradation process. So the specific region of bacterial genome must be activated for degradative enzymes to be produced. And if you see the types of enzymes, there are two types of enzymes. One is constitutive enzyme and one is inducible enzyme. You can uh, you can view and differentiate these enzymes. I will give one instance like you can compare constitutive enzyme as a permanent employee in an office, whereas inducibles are contact based employees. So what happens? Permanent employees, they will be always present in the office throughout the year, whereas contract employees, they work for specific period of time and whenever the project needs to be completed, they recruit extra persons in order to complete the work. So this is same way in the constitutive enzymes, the microorganism produce these enzymes continuously for running their central metabolic process, that is, for their energy production or for their growth or for their basic metabolism. Whereas the inducible enzymes, what happens? Here, based on the toxicity, their genomes are switched on or off and they secrete enzymes that can break down the pollutant. Right. So here what happens whenever the microorganism needed, they will induce this enzyme and initiate the bioremediation and they are generally inducible. So this is the major difference between the constitutive and inducible promoter. So let's take on an example. Think of all hydrocarbons that are present in the soil. So if the soil bacteria encounters toluene, one of the hydrocarbons, it doesn't always have the enzymes ready, right? So what happens? Instead, the presence of if the particular microorganism has the capacity to consume the toluene, and in the presence of toluene, it can act as a light switch. So whenever there is a presence of toluene, they will secrete specific gene that produces the toluene dehydrogenase enzyme, which are capable of degrading this pollutant. So by this, we can ensure that energy and resources are not waste when the pollutant are not around. So what does it tells us? Whenever the microorganism are in the toluene environment, they only switch on the genomes that are capable of producing the particular enzyme. So so whenever these enzymes are not needed, they don't produce. So this is going to save lot of energy to the microorganism. So which can be used for other purposes. So the last principle is the availability of the appropriate electron acceptors and electron donors. So as we all know that the electron acceptor is nothing but it accepts the electron in order to complete the bioremediation process. Whereas donor means it donates the electron to complete the process. So generally the microorganism will obtain the energy through oxidation and reduction mechanism where the electrons will be transferred from one substance to the another substance through electron transport chain. So what happens here? The microorganism will try to oxidize the organic pollutant, for example, the hydrocarbons into simple compounds like CO2 and H2O. While doing this, simultaneously the electrons will be released and they pass through the different electron acceptors. So in case of aerobic mode of respiration, they use oxygen as an electron acceptor and they convert into water. Whereas in the anaerobic mode, they use nitrates, sulfates, or carbon dioxide in order to complete the process. So these microorganisms will try to oxidize the pollutant to CO2, achieving a biomineralization strategy. At the same time, they tend to reduce the electron acceptors. In the case of aerobic process, molecular oxygen will act as an electron acceptor, and in case of anaerobic process, nitrates and sulfates will be acting as an electron acceptor to complete the process. So these are all the five most important principles which determines the rate of degradation. So at the end, we have to understand that it is not only the right microorganism, the right microorganism needs to produce right enzyme in a right environmental condition with an appropriate electron acceptor. And moreover, the bioavailability is also an important factor which can turn the toxic environment to the non-toxic and harmless product. So these are all the five most important principles that we have to keep in mind while studying the bioremediation process. So as we have seen, most of the bioremediations are energy yielding biochemical reactions, and most of them are oxidation in nature. Oxidation means they tend to lose the electrons. So the microorganism will tend to oxidize these organic pollutant into product, and they transfers these electrons to the electron acceptor, which tends to reduce. So once again, in the absence of molecular oxygen, anaerobic uses other form of electron acceptors like nitrates, sulfates, CO2, etc. And if you see the hierarchy of accepting the electron transport, the most convenient way is the oxygen as an electron acceptor, followed by the nitrate, followed by manganese, followed by the iron, followed by the sulfate and hydrocarbonates. So these are all the hierarchy, and depending upon the environment, whether it is an aerobic or anaerobic, the electron acceptors varies from microorganism to the microorganism. And if you see the factors affecting the rate of biodegradation, the first is their availability of nutrients. And if we see the nutrients, there are two major groups of nutrients, which is micronutrient and macronutrient. And if you see the micro and macronutrient, the term itself gives an idea that the macronutrients which are required in the large quantity. Why it is required in a large quantity? Because it helps in building block to constitute these cells and enzymes. That is why they require in the bulk quantity. The examples of macronutrients are, the first is nitrogen. Why it is used? The nitrogen can act as an protein which are used for synthesizing the amino acids and enzymes. Whereas phosphorus, it helps to produce ATP and DNA. And in case of sulfur, it acts as an coenzyme for carrying out the biochemical reactions. In case of calcium, it helps to strengthen and stabilizes the cell wall. Whereas magnesium, it helps to stabilize and hold the ribosomes. And in generally, the microbial growth in the subsoil, they are not limited by nitrogen and phosphorus, as long as the contaminant concentration are present in very minute amount, which is ppm level. In 1 liter, it will be present in some mgs of pollutant. And in case of aquifers, the carbon nitrogen phosphorus ratio of 30 is to 5 is to 1 is normally observed, and it is sufficient to ensure the unrestricted or undesired growth in case of aquifers. So, an equal importance of macronutrients is that another term is micronutrients. The term itself telling us that these nutrients are required in very small amount. For example, the vitamins, minerals, they require in very tiny amount, and some trace metals, especially for specialized enzymes, they act as an cofactor or coenzyme factor in order to catalyze the speed of the reaction. And iron, sometimes without this, the microorganism can't run the energy machinery. So every microorganism need the nutrient, both the macro and micronutrient. So it has to be present in the balanced state in order to catalyze the reaction. And the other factors, environmental factors, are, as we already discussed, the pH and the buffering condition, which is the narrow pH range of 6 to 8 is mostly preferred by the mini microbes, and in especially with the aquifer microbes, it can well perform between the 5 to 9. And buffering capacity of carbonates and silicate minerals of the aquifers play an important role. So always optimum pH is required for carrying out the microbial process. And with regard to the temperature, it helps in catabolizing the microbial metabolism. That is, they speed up the reaction to a particular point, and after that it starts to decrease the rate of reaction, and we call that particular temperature as optimum temperature, where the growth of the microbial community will be very higher. And below it, it became inactive. And for most of the bacteria, it prefers the temperature around 20 to 40 degrees Celsius. And below the temperature, it often slows the microbial growth. And above it, it eventually kills or doesn't survive at the very high temperature. But exceptions are there. If we see the high or thermophilic microorganism, their optimum temperature is at a very high temperature. Whereas in case of saccharophilic microorganism, their optimum pH will be in the lower temperature. So based on the temperature, based on the microorganism, the temperature will vary, and we have to find the optimal temperature. And the third most important factor is that absence of toxic or inhibitory substance. So this is the last and most important factor, because even if the conditions are right, some toxic element can spoil everything. So this toxic pollutant can inhibit the microbial activity and the biodegradation. So what happens? For example, the heavy metal, even above 1 ppm, some heavy metals like cadmium and chromium, they act as an toxic in nature. They inhibit the growth. And sometimes even the byproduct of the microbial action became the toxic, like the H2S from the SO4 reduction. They act as a toxicity if their concentration is present at 200 mg per liter, which we call it as 200 ppm. And the substrate utilization. So if microbes are given an easier food like sugar, it will assimilate these easily. They assimilate easily and ignore the order complex substance. So this is why the presence of easily degradable substance can inhibit the degradation of the target pollutant. So it is always preferred to uh, identify the microorganism that can use the carbon source as the polluter. So from these factors, we understood that right pH balance, right temperature, and free of toxic or inhibitory substance, helping us to turn the pollutant into a clean and healthier ecosystem. So this is why you need to understand the various aspects, various factors of the bioremediation approaches. So So these are all the content that I want to discuss in this session. And we have seen the bioremediation, five principles of bioremediation, and we have seen the factor affecting the biodegradation process and many more. So this is all about the biodegradation. And if you have any doubts, you can post your doubts in the chat box. And if you do not have any doubts, we can move on to the MCQ session without any delay. So any doubt? Can we move to the MCQ session? Okay, then we will move on to the MCQs now. So the first MCQ is the plastic waste at a landfill was found to harbor a population of bacteria naturally capable of breaking it down. So addition of nitrogen-rich organic waste was observed to accelerate this process. So this is an example of option A, biostimulation, option B, bioaugmentation, option C, composting, option D, bioreactor, option E, landforming. So I will explain the question once again. So you have a plastic waste at a particular landfill, and in that landfill you already have the some microbial community, that is bacteria, that has the capability to degrade this plastic waste. So we are not going to introduce any microorganism here. We are going to add the nitrogen rich organic waste. So what do we call this strategy? If we are adding the if we are adding the nutrients to the environment to speed up the degradation process, whether we call this as a biostimulation or bioaugmentation or composting or bioreactor or landforming. You can post your answers in the chat box, or you can unmute your mic and you can answer the question. And your answers are your answers are option A. Okay. Any other answer? Option A, biostimulation. Okay. Any other answer? Okay. The correct answer is yes, it is option A. And why it is? Because biostimulation is nothing but we will be adding the nutrient. Like in this case, we will be adding organic waste to the environment to enhance the activity of the existing microbes that can degrade the contaminant. In this case, the plastic waste. So we are adding only the nitrogen rich organic waste. That is why it comes under the concept of biostimulation. And what about the other options? What do you mean by bioaugmentation? So in bioaugmentation, what we do is we'll be adding the microbial population to the contaminant sites to degrade or break down completely. Whereas in composting, it is a controlled process of decomposing the organic materials like the food waste into nitrogen into gas. So in a nutrient rich environment. So the option D, bioreactor. A bioreactor is a technology which is designed to optimize the microbial degradation of pollutant under the controlled environment. The controlled environment is nothing but the controlled pH, temperature, and oxygen level. And final landforming, where the contaminated soil is spread onto the land and exposed to the natural weathering and microbial degradation. So this is a long process and it takes years to remediate it. So from the question, biostimulation is the right answer. So now we will move on to the next question. One of the oldest bioremediation system is the activated sludge system. Here, a fraction of solid waste containing the microorganism and partially treated waste is added to the new untreated waste in a wastewater treatment plant. So this is best described as, read the question. In the activated sludge system, a fraction of solid waste which contains both microorganism and the partially treated waste. It is added, this activated sludge is now added to the wastewater treatment plant in order to treat the pollutant. So this describes which one of the following category. Option A, in situ intrinsic process. Option B, ex situ intrinsic process. Option C, in situ engineered process. Option D, ex situ engineered process. So among this, which best suits for the above process. I will read the question. One of the oldest bioremediation system is activated sludge system. Here, a fraction of solid waste containing the microorganism and partially treated waste is added as to new untreated waste in the wastewater treatment plant. This is best described as, you can post your answers. And your answers are option C. Okay. Now we will see the answer. It is option D. Now we will see why it is option D, which is ex situ and engineered process. And why it is ex situ, we will see now. Ex situ means we will be taking the contaminants inside an artificial enclosure. Here, what we are doing, we are taking the fraction of solid waste and we are treating in the wastewater treatment plant. Wastewater treatment plant is normally observed in the industries. So, here what we are trying to do is we are taking the waste from the one environment and we are treating in the another environment. That is wastewater treatment plant. That is why this process is called ex situ. The activated sludge system removes the waste from its original location and involves active human intervention and control to create the optimal environment for the microorganism, making it engineered process. So here we are treating in the wastewater treatment plant. We are not treating the pollutant in the normal environment or at the original site. That is why it is an ex situ engineered treatment process. And in situ, as we already know, it is a bioremediation occurs in the original location of waste. Whereas intrinsic means it is a process that naturally occurring microorganism and condition that occurs any without human intervention or modification. Whereas if you see the activated sludge segment, it is highly controlled and manipulated process to enhance the biological activity. That is why it is an engineered and we are taking this pollutant outside from the site. And that is why it is called as waste or that is why it is called as ex situ engineered as the processes occurring in the external environment, that is wastewater treatment plant. So we will move on to the next question. Bioremediation 2.0 refers to the use of option A, purified enzyme, option B, bioengineered enzymes, option C, bioengineered microorganism, option D, pure bacterial culture, option E, group of microorganism. So I will repeat the question. Bioremediation 2.0 refers to the use of option A, purified enzyme, option B, bioengineered enzyme, option C, bioengineered microorganism, option D, pure bacterial culture, option E, group of microorganism. You can post your answers. bioremediation 2.0 refers to? Okay, option C. Now we will see the answer. Yes, it is correct. Option C, bioengineered microorganism. And what do we call the bioremediation 2.0? It is an engineered whole microbes or it is a super bugs with required pathway and enzymes for in situ pollutant mineralization. So it is an engineering the whole microbes. And if we see the other options, as we understand, it is an engineer the whole microbes. It is not denoting just the enzymes. So these options will be eliminated. And here we refer to whole microorganism. So the bioengineered enzymes will also not be a part of bioremediation 2.0. And if we see the genetic makeup of bacteria to enhance the pollutant degrading capability is not just the pure and unmodified bacterial culture. It needs to be genetically modified. And the group of microorganism we call that as consortia, which is also not regarded as bioremediation 2.0. So anything which is bioengineered using genetical tool, we call them as bioremediation. Rest all are not the best option that suits for bioremediation 2.0. Yes, your answers are correct, and we will move on to the next question. Which one of the following is included in pre-genomics molecular analysis approach? Option A, meta barcoding analysis. Option B, metagenomic analysis. Option C, meta transcriptomic analysis. Option D, 16S or RNA based identification. So, which one of the following is included in the pre-genomic molecular analysis approach? And your answers are option B, okay, option B and option D. So, any other answer? Option D, okay, B and D. So now let's see the answer. The correct option is option D. Why it is? First, the pre-genomics refers to the molecular analysis technique that predate the advent of modern genomic sequencing technology. That is, before the modern genomic sequencing technology, we call all the technology as pre-genomics. That is, these methods typically focus on specific genes or small region of the genome. Pre-genomic methods focuses on specific genes or small region of the genome, while post-genomic involves sequencing these entire genomes or transcriptomes. So if you see the among all other options, 16S rRNA sequencing predated the modern genomic sequencing. If you see the other options, the first one is metabarcoding. What happens? It uses the DNA as a barcode to identify the species that are used conjunction with the next gens sequencing. And these next gens are all post genomics molecular analysis. And metagenome is nothing but it will sequence the entire DNA content. So that is why it is also considered as a post genomic molecular analysis approach. And if you see the option C, meta transcriptomic analysis, so it will sequence the RNA transcript from the sample. So this is also obviously going to be an post. So the first three options belong to post genomic molecular analysis approach, whereas the 16S rRNA only fits with the pre-genomic molecular analysis approach. So rest are all post genomic molecular analysis approach. So we will move on to the next question. The next question is the bacterial strains was genetically engineered to break down the xylene and showed promise in the laboratory studies. After the engineered strain was added to the river contaminated with the xylene containing the industrial effluent, the xylene level remained unchanged. Which of the following is not a possible explanation for this observation? So I will try to explain the question. So what happens is one scientist they are trying to engineer a particular bacterial strain which is designed for breaking down the xylene. But in the laboratory condition, the xylene has degraded drastically. But with the same strain, when the scientist was applying to a real-time wastewater, that is industrial effluent, the scientist was not able to observe the same degradation pattern. And among these statements, which is not a possible explanation for this observation. So the option A, the ambient pH and temperature of the habitat may be unsuitable for the bacteria. Option B, the resident microbial community may be resisting the colonization by externally added bacteria. Option C, there may be a lack of other nutrients that are required for growth and bioremediation. And option D, the genetically modified bacterium lost its xylene degrading capability due to lack of selective pressure. Option E, co-pollutant in the industrial waste may be toxic to the bacterium. So among this, which is not a possible explanation for this observation, you can post your answers. And your answers are which among the following statement is not a possible explanation? Option D. Okay. Option D. The genetically modified bacteria could have lost its xylene degradation capability due to lack of selective pressure. Okay. We will see the answer. Yes, it is correct. Option D is not the possible explanation because as the scientists have already selected the toxic xylene itself as the selective pressure. This is not the possible correct answer for this observation. And why the other options are correct? Because in the first condition, the ambient temperature and pH of the habitat may be unsuitable for the bacteria. This might be true because, as we have seen in the lectures, the environmental condition significantly influences the microbial growth and activity. So this could be the possible reason for this observation. Whereas in the option B, the resident microbial community may be resisting the colonization by externally added bacteria. Yes, this might be also a reason because introducing the microorganism into an real-time environment faces the competition and potential inhibition from the existing microbial community, so which hinders their establishment and the effectiveness of biodegradation. So this could have also be an reason. And option C, there may be a lack of other nutrients that are required for growth and bioremediation. Yes, because microorganism requires other nutrients. We already seen micro and macronutrients for their growth and carry out their bioremediation process. Option E, co-pollutant in the industrial waste may be toxic to the bacteria. Yes, the presence of other pollutant can also be a toxic to the engineered bacteria, which reduces the viability and ability to degrade these xylenes. So option D is not the possible explanation for this observation. We will move on to the next question. Aerobic biodegradation of organic pollutant primarily occurs through oxidation. Option A, option B, reduction, option C, gasification, option D, activation, and option E, inactivation. So, aerobic biodegradation of organic pollutant primarily occurs through. You can post your answers, and your answers are your answers are option A. Okay. Any other answer? Aerobic biodegradation of organic pollutant primarily occurs through option B, reduction. Okay. So now we will see the answer. Correct answer is option A, oxidation. Why it is so? Aerobic biodegradation means breaking down the organic pollutant in the presence of oxygen. What happens here? The microorganism will use the oxygen as an electron acceptor and they oxidizes the organic pollutant into simpler ones, often involving addition of oxygen atom to the pollutant. So they are trying to oxidize the organic pollutant. Here they are asking for organic pollutant. So the organic pollutant tends to oxidize. If they ask for oxygen, they tend to reduce. So organic pollutant get oxidized. So the correct answer is oxidation. What about the other options? Reduction means it is directly opposite to the oxidation. Here what they gain the electron and they reduce oxygen into water. And if it is an anaerobic condition, they use other modes of electron acceptor. Gasification means it is an high temperature process which converts the organic pollutant into gases like CO2, H2, etc. So it is normally not a biological process and doesn't involve any action of microorganisms. So this option will be eliminated. And activation, what does it mean? It means like it makes a molecule more reactive by adding the energy or changing its chemical reaction. Whereas inactivation is just opposite to that of activation. It tries to help break down the pollutant and making inactive or ineffective. So among this, the more appropriate answer is the oxidation, where oxidation of organic pollutant takes place in the aerobic mode of respiration. So we'll move on to the next question. DAS is the most abundant potential electron acceptor for the oxidation of organic pollutant under anaerobic condition. Option A CO2, option B H2, option C N2, option D Fe3, and option E Fe2. So, which is the most abundant potential electron acceptor for oxidation of organic pollutant under anaerobic condition? You can post your answers. Your answers are okay. Option D, option A, okay. Option D, option A, and option D. So let's see the answer now. The correct answer is option D, Fe3. Why it is so? Because Fe3 addition stimulates the anaerobic bioremediation process considerably when compared to other electron acceptor. And if you see CO2, the option A, it is an end product of the anaerobic process, right? So if you see the thermodynamic progress of electron acceptor, you will observe CO2 is the last electron acceptor. And in case of H2, it is a common electron donor observed in the anaerobic condition. So it is not an electron acceptor. Whereas N2, it is a stable form of nitrogen molecule. So it is not directly as electron acceptor seen in anaerobic condition. And Fe2, it is a reduced form of ion resulting from the Fe3. So it is not an acceptor itself. So among this, Fe3 is the most abundant potential electron acceptor for the oxidation of organic pollutant. So we'll move on to the next question. Uranium bioremediation involves its utilization as an energy source, utilization as an electron source, utilization as an electron sink, option D methylation, option E acetylation. So uranium bioremediation involves option C, okay, utilization as an electron sink. Any other option? Any other answer? Option B, okay. Let's see the answer. It is option C, utilization as an electron sink. Why it is so? Because uranium 6 is reduced to uranium 4 and they are precipitated. This makes it less bioavailable and reducing its toxicity. And if you see the other options, microorganism normally uses the organic compound or other readily available electron source. But in this case, it is uranium. It doesn't use it as energy source or electron source. So uranium cannot be used as a primary electron or energy source. And if you see the methylation, what does it means? It is a process which modifies the structure of the organic compound. While some microbes can methylate the uranium, but it is not the primary mechanism involved in it. Likewise, acetylation, it is not a significant mechanism observed in the uranium bioremediation. See, the more appropriate answer is it is utilized as an electron sink rather than an energy or electron source. So we will move on to the next question. The Geobacter metylar reducens is a motile microorganism. Its motility was not discovered until it was subjected to option A, pure culture isolation, option B, hanging drop method, option C, microscope, option D, whole genome sequencing, option E, metagenome sequencing. So I will repeat the question. Geobacter metylireducens is a motile microorganism. Its motility was not discovered until it was subjected to. You can post your answers now. Option B, option D, option B, and option D. Now we will see the answer. The correct answer is option D, which is whole genome sequencing. So what happens here is, so this bacteria is a motile microorganism, but it does not express its flagellar genes under normal culture condition. So hence its motility was not discovered until whole genome sequencing revealed the presence of that concerned motility flagella genes and prompted the modification of cultural condition to trigger their expression. So under normal lab condition, it doesn't show any motility. And we all know that option A, B, C, these are all conducted in the laboratory condition. So what happens in the pure culture isolation? The isolation allows the motility observation only if flagella is present. But here, their flagella was not distinctly visible. So option A is not correct answer. Whereas option B, hanging how drop method, it observes the motility in the small drop of liquid. So as the question itself says that under their motility was not discovered, right? So option C is also wrong, whereas if we see the option E, metagenome, it collects the DNA from the community and which tells us the information about the genetic diversity, not the phylogenetic characteristics of the individual organism. So whole genome helps us to find the presence of this flagella gene. This led to the identification of motility later. So that is why the whole genome sequencing, when subjecting to this, they identify it as an motile organism. So the last question for the today's session is, today the primary limitation on genomic analysis is option A, the lack of high throughput sequencing techniques. Option B, the lack of adequate culturing techniques. Option C, lack of DNA extraction efficiency from dirty samples. Option D, lack of cloning library preparation efficiency. Option E, lack of methods with adequate sequencing depth. So among this option, which is the correct limitation, your answer, you can post your answers. Today, the primary limitation on genomic analysis is lack of high throughput sequencing techniques, or lack of adequate culturing techniques, or lack of DNA extraction efficiency from dirty samples, or lack of cloning library preparation efficiency, or it is a lack of method with adequate sequencing depth. Option B, okay. Any other answer? We will see the answer now. The option B, the lack of adequate culturing techniques, because by definition, genome sequencing requires a pure culture. As of now, most of the microorganisms are remain unculturable. And from the earlier lecture, we understood that 98% of the cultures are unculturable. So there should be culturing techniques needs to be developed. This is the primary limitation when compared to all the techniques. So why not other options? If you see, if you see the high throughput sequencing, as we already know, there are a lot of NGS, that is new next gen sequencing has been developed, which is rapid and high throughput sequencing. So this is not a primary limitation today. And if you see, there is a lot of DNA extraction efficiency from the dirty sample. That is, this is not a primary bottleneck considering the advancement in the sequencing technology. And more importantly, we all know that the many microorganisms cannot be cultured in lab, which hinders the study of their complete genome. That is why the option B, the lack of adequate culturing techniques, play an important role in limitation on genomic analysis. So this is for all in this week, and thank you once again for patiently attending the lecture. If you still have any doubts, you can mail me in the Gmail, ajitweli284@gmail.com. You can post your queries in the NPTEL Environmental Forum, discussion forum, so that our team can address the queries. And if you want to revisit these videos, you can go to the YouTube's or NPTEL Environmental Technology tab, where you can access the recording sessions as well as the PPT. So this is all for the today's class. So the next week we will continue the main application part of bioremediation in the real-time condition. So thank you all. Have a great day. If you have any question or any doubts, you can post. Okay, thank you. Thank you, Ashwini. Thank you, Tuhin, for attending the lecture. So I think we can wind up the session. Thank you all.